Good morning, students. Today I'll be talking on a very important and interesting topic, that is the cell cycle. This is wonderful how a cell divides and how a cell is going to divide and who will control this. So, every cell must have an ancestor from which it arises. A German pathologist, Rudolf Virchow, in 1858 proposed a cell doctrine. And this doctrine states that cells are generated from cells, and the only way to make more cells is by division of those already exist. A cell reproduces by performing an orderly sequence of events by which the cell duplicates its contents and then divides into two. This cycle of duplication and division is called the cell cycle. Details of cell cycle were given by A. Howard and Peck in 1953. In unicellular organisms, like a very small organisms like bacteria and yeast, each cell division produces a complete new organism. This is very wonderful. But in case of us, like multicellular organisms, higher plants, including human being and other many animals, long and complex sequences of cell divisions are required to produce a functioning organism. So, cell division is also required even in the adult body to replace the cells that die. The details of cell cycle vary from organism to organism and at different times in an organism's life. Eukaryotic cells have evolved a complex network of regulatory proteins known as the cell cycle control system, which governs progression through the cell cycle. The core of this system is an orderly series of biochemical switches that control the main events of the cycle, including like DNA replication, then segregation of the replicated chromosomes. Duration of cell cycle is variable. It may take 10 to 20 hours or more in most growing plant and animal cells, and it is 24 hours in case of rapidly dividing human cell. Whole life in nerve cells and island cells. So the present topic is primarily concerned with how the various events of the cell cycle are controlled and coded. So in this connection, we can just have certain phases of the cell cycle. The cell cycle is completed in two major phases. Number 1, interphase, and number 2, mitosis. The interphase is completed again in the following steps, like G1 phase, S phase, and G2 phase. G1 phase. G stands for gap. It is the most variable period. It may last days, months, or years, depending on external conditions and extracellular signals from other cells. In human being, it takes about 9 hours. If extracellular conditions are unfavorable, cells delay progress through G1 and may enter G0. Indeed, many cells remain permanently in G0 until they or the organism dies. If extracellular conditions are favorable and signals to grow and divide are present, for example, we have epidermal growth factors, we have fibroblast growth factors, lymphokines, nerve growth factors, etc. Then cells in early G1 or G0 progress through a commitment point near the end of G1. So we call it start. For example, in yeast, but it may call restriction point in case of mammalian cells, and enter S phase. During this phase, there is synthesis of RNA, proteins, and other biomolecules such as nucleotides, carbohydrates, lipids, etc. Cells also increase in size. G1S checkpoint ensures that the cell is ready for DNA synthesis. So now let us look about the S phase. S stands for synthesis. During this particular phase, the synthesis of DNA takes place, and thus the amount of DNA becomes double. The synthesis of RNA and protein also continues, especially histone formation takes place. In human, it takes about 10 hours. And next is G2 phase. The DNA synthesis stops during this phase. However, synthesis of RNA and protein continues, including the synthesis of microtubular proteins like tubulin. Mitochondria and chloroplasts also divide. Then centriole duplication begins in G1 and is completed by G2. Biogenesis of other cell organelles takes place. The cells also continue to grow. Duration in human being is about 4.5 hours. So now we have mitotic phase, and commonly we call it M phase. The M phase of the cell cycle is the culmination of the cycle, and it includes the various stages of nuclear division like karyokinesis and cytoplasmic division called cytokinesis. The central problem for a mitotic cell is how to accurately separate and distribute or segregate its chromosomes, which were replicated in the preceding S phase, so that each daughter cell receives an identical copy of the genome. W. Fleming in 1892 coined the term mitosis. So now we shall be again having about the different phases of mitotic phase or mitosis. First, prophase, metaphase, anaphase, telophase. First, prophase. The first readily visible sign that a cell is about to enter M phase is the progressive compaction of the replicated chromosomes, which become visible as thread-like structures, and the process is called chromosome condensation. There are certain proteins called condensins which perform the work of chromosome condensation. Ardiphophase. During ardiphophase, the cell becomes spheroid, more refractile and viscous. Pair of centrioles migrates toward the opposite poles. Formation of esters, that is amphiestal spindle in case of animal cells and anestal spindle in case of the plant cells, takes place during this particular phase. Then metaphase. The chromosomes shift toward the periphery or nuclear envelope, leaving a clear central area in the nucleus. At the same time, further condensation of chromosomes occurs, and each chromosome appears to consist of 2 chromatids. The sister chromatids are glued together by multi-subunit protein complexes. We call them cohesins, and these are deposited along the length of each sister chromatid. Primary constrictions or centromeres become visible during this particular phase, and these kinetochores are complexes of proteins that assemble onto the highly condensed DNA at a centromere. Then coming to late prophase, it is sometimes called prometaphase. The nucleoli and nuclear envelope become disappeared during this particular phase, and at the same time the spindle fibers are also formed. The chromosomes become attached to the spindle fibers through their kinetochores. The attachment of chromosomes to the spindle is a dynamic process, and it seems to involve a search and a capture mechanism in which microtubules nucleate from each of the rapidly separating centrosomes, which grow outward toward the chromosomes. Now we have metaphase. One of the most striking aspects of metaphase in vertebrate cells is the continuous oscillatory movement of chromosomes at the metaphase plate. Spindle fibers that connect the chromosomes are called chromosomal fibers, and those that extend without interruption from one pole to other are called continuous fibers. These continuous fibers also appear at the same time. Smaller chromosomes are usually central in position, whereas the larger ones are peripheral. The chromosomal movement toward the metaphase plate is caused by kinetochore and a polar microtubular interaction. During metaphase, an equilibrium of forces is established. There are pole-directed forces that tend to pull the sister kinetochores toward the poles. Also, polar microtubules exert a counteracting force that tends to pull the poles inward. The next phase is called anaphase. So anaphase begins abruptly with the release of cohesion linkage that holds the sister chromatids together at the metaphase plate. The metaphase to anaphase transition is triggered by the activation of the anaphase promoting complex. We call it APC. Once this proteolytic complex is activated, it has two crucial functions. One, it cleaves and inactivates the M phase cyclin, thereby inactivating mCDK. And two, it cleaves an inhibitory protein called securin, thereby activating a protease called separase, which cleaves a subunit in the cohesion complex to unglue the sister chromatids. Now, the sister chromatids are separated from each other, and each chromatid migrates to its opposite pole. The chromosomes move by two independent and overlapping processes. The first, called anaphase A, is the initial poleward movement of the chromosome, accompanied by shortening of the kinetochore microtubules at their attachment to the chromosome, and to a lesser extent by the depolymerization of spindle microtubules at the two spindle poles. The second process, called anaphase B, is the separation of the poles themselves, which begins after the sister chromatids have separated and the daughter chromosomes have moved some distance apart. Anaphase A depends on motor proteins at the kinetochore. Anaphase B depends on motor proteins at the poles that pull the poles apart, as well as on motor proteins at the central spindle that push the poles apart. Chromosomes may exhibit different shapes depending on their location of centromeres. So they may be V shape, they may be L shape or J shape. The chromosomes usually move toward the poles at the rate of about 1 micrometer per minute. Now we have the last phase called telophase. During telophase, chromosomes start to unfold and become less and less condensed. That is what we call it, decondensation of chromosome. The new daughter nuclei are formed. At the same time, the esters and spindle fibers are completely disappeared. Now cytokinesis takes place. Different cytoskeletal structures are responsible for cytokinesis. In many animal cells, a contractile ring that contains both actin and myosin filaments forms around the equator of the cell, just under the plasma membrane. As the ring contracts, it pulls the membrane inward, thereby dividing the cell into two. Cytokinesis is centripetal in animal cells and centrifugal in plant cells. In plants, there is formation of cell plate between the two daughter nuclei. The cell plate represents the middle lamella between the walls of two adjacent cells. The sudden transition from metaphase to anaphase initiates the dephosphorylation of many proteins that were phosphorylated at prophase. So lastly, we shall be having that how this cell cycle is controlled, that is the control of the cell cycle, or we can say regulation of cell division. The cell cycle control system consists of various cyclin-dependent kinases. We call them CDKs. The CDKs become activated when regulatory proteins called cyclins, also called CDK regulators, bind to them, as well as by phosphorylation and dephosphorylation of kinase. Several classes of human cyclin have been identified. They are cyclin D, E, A and B. Cyclin D and E function during G1 phase, and therefore they are collectively referred to as G1 cyclins, whereas cyclin E is mainly involved in G1 to S transition, and therefore it is called G1S cyclin. Cyclin A is mainly involved in S phase, and it is called S cyclin. So this cyclin A and B are involved in G2 to M transition and early M phase. And since cyclin B mainly takes part in G2 to M transition, cyclin B is also called G2M cyclin or simply M cyclin. Mammalian CDKs are CDK1, 2, 3, 4 and 5. These are serine threonine protein kinases, which become active when cyclins bind to them. When cells in G0 are stimulated with growth factors, cyclin D appears first. CDK2, CDK4 or CDK5 cyclin D complex helps transcription of various genes for other cyclins and many enzymes. Cyclin E binds to CDK2, and this CDK2 cyclin E complex pushes the cell from the G1 to the S phase, whereas CDK2 cyclin A complex controls the events of S phase. CDC2 cyclin A or CDC2 cyclin B are involved in G2 to M transition and early M phase. CDC2 is the catalytic subunit of MPF, that is mitosis promoting factor. Breakdown of nuclear envelope requires depolymerization of the nuclear lamina, and this depolymerization is a direct consequence of phosphorylation of nuclear lamine proteins by MPF. Phosphorylation of chromatin proteins by MPF is thought to cause the condensation of mitotic chromosomes. Similarly, MPF-induced phosphorylation of microtubule-associated proteins is thought to cause the dramatic reorganization of microtubules into the mitotic spindle apparatus. The onset of anaphase is induced by degradation of mitotic cyclins. Degradation of mitotic cyclins inactivates MPF, and as a result of this, the sites in various proteins that were phosphorylated by MPF are dephosphorylated, leading to the late mitotic events. For example, dephosphorylated nuclear lamine proteins repolymerize into the nuclear lamina of the two daughter nuclei during the late telophase. Chromosome decondensation is thought to result from dephosphorylation of chromatin proteins. Similarly, dephosphorylation of microtubule-associated proteins is thought to be responsible for deassembly of the mitotic spindle by returning microtubule dynamics to those of an interphase. Finally, Dephosphorylation of the inhibitory site on cortical myosin initiates contraction of the cortical ring, causing cytokinesis and cell separation. Checkpoints control operations to ensure that each phase of the cell cycle is completed before the ensuing phase begins. Now we have conclusion. Cells are arise from cells through division. Mitosis represents only a small part of the life cycle of a cell. The cell spends most of its lifetime in interphase, the period during which it doubles in size and replicates its DNA. Thus, the cell cycle is completed in two major steps. One, interphase, another is mitosis. Interphase includes G1 phase, S phase and G2 phase. During mitosis, the chromosomes try to undergo specific arrangement by which they first move to equatorial plane and then separate their sister chromatids, which finally move to opposite poles, resulting in the formation of two daughter cells having the same genomes. The cell cycle is controlled by various cyclin-dependent kinases, CECs, which become activated when cyclins bind to them, as well as by the phosphorylation and dephosphorylation of kinase. When the cells in G0 are stimulated with growth factors, various cyclins appear and CECs become active, and thus the various events of cell cycle are regulated. So that's all. This is about how a cell divides and how a cell is ready to divide and who will control it. So thank you very much for your patience.